All right, let's get busy. So, good morning, everybody. What we're going to be doing today is going over the intro PowerPoint, which is really kind of terminology and language. And then we'll be getting started on the flow of genetic information, which is essentially some kind of recap material, which will or should get you up to speed from uh, whatever you've studied before genetics. Or if you've come into genetics after not having studied for a long time, then uh, giving you some good material to kind of, you know, dive back in, essentially. All right, so let's see, let's share the screen. Let's do this one first. Where are we at? Here we go. So this is really a just like an overview PowerPoint, right? There's not a whole bunch in here which you really need to worry about in terms of what's going to be on the exam and stuff like that, because it's not really a kind of an information type PowerPoint. It's really something to give you some language and some kind of perspective, I guess. Right. So genetics means a lot of things to different people, right? So you can think about genetics in terms of why do I look the way I am? Why do I look a little bit like my mother, a little bit like my father, and not exactly like either of them. Right? You can think of it in terms of a particular familial trait. So we've got the Crook family chin, which you can't see right now because it's hidden by a bunch of fur. Crook family nose, right? And if you look back through pictures over the generations, you'll see those particular characteristics, right, popping up every now and then. So clearly there's something heritable there, which I inherited from my parents and they inherited from theirs. You could think of it in terms of, say genetics to someone who's currently receiving treatment, for example. They may think of genetics in terms of why is this drug working well on me or why is it not working on me, right? What about my personal genome dictates the response to this drug? So we're talking about like the molecular genetics, right? And that's something that is of huge concern in the pharmaceutical industry, and it's actually a big thing right now in this field called personal genomics, right? Which is another way of thinking about this, right? So it's not just about one or two genes, but more about your whole genome, right? And how different patients respond to medicine. So I don't know about you, but let's get chat back up just in case anyone asks something. But personally, I never take ibuprofen for pain or headaches or anything like that. It just doesn't work. How was it called? Naproxen, I think, which is like the, dam busters of the ibuprofen world. Those have a small effect, but they just don't do anything, right? And so whatever is going on in my liver, most likely, it either metabolizes ibuprofen way faster than other people, so it never gets the chance to do its job, or it doesn't metabolize it and turn it into active form, right? So there's kind of two different perspectives. I don't actually know which way around it is, ibuprofen. I think it's inactivating ibuprofen that's the issue, right? So that's both a span of molecular genetics, right? There are clearly enzymes in my liver, right, which are encoded by genes in my genome, which do a really, really good job of breaking down ibuprofen, right? But also, there's probably lots of other things that affect that too, right? So it's also the the big scale, at least as an individual, of my collective genome, right? What else? There's also, if you talk to biologists in general, but particular ones, and you say genetics, they'll be thinking really about why is this species, and why is it distinct from a related species, right? What changes occurred over a long period of time, which resulted in these two different groups of whatevers, could be birds, could be sea slugs, could be nematodes, right, which is close to my heart, to result in these two groups being reproductively incompatible, right, which is one of the standard definitions of being a species. What happened over a long period of time, essentially. And so really, that's kind of evolutionary genetics. And then if you're kind of a doctor, maybe you're in the research field, and you're responsible for treating, I don't know, let's say cardiac disease, right? Well, when you look at your population of individuals, what is their varying predisposition to cardiac disease? So if you look at, let's say you're a doctor in Oshkosh, Wisconsin, right, which is way up in the north. I think it's just stopped snowing there, more or less. I don't know, that's a joke. But anyway, almost certainly you're going to have an almost entirely white population, right? And so the propensity for heart disease is going to be a particular rate or a particular type of heart disease, maybe strokes versus heart attacks or whatever, right? Now you look at, let's say, swamp country, Louisiana, right? You're going to have a very different makeup in the population, right? You're going to have many more Black people. You're going to have some Hispanics, but not too many, right? That tends to increase more as you get west towards Texas, right? And so depending on their, evolutionary history, and also, the fact that they're like frying everything, you're going to have a different potential predisposition for heart disease, right? And so in terms of genetics, really what we're talking about is what is the population, right? What's the genetic makeup of that population, and how does that change depending on different factors, right? People coming in, people leaving. If you look at San Antonio, right, it's majority Hispanic, 55, 60%. I don't know the percentage of African Americans, I think around 10%, give or take, and the rest white. But there's a huge flux of people coming in from different parts of the US, from Mexico, I'm not sure from Canada, but you've got a very different makeup than if you go just a little further outside of San Antonio, or maybe further west to like El Paso, or south towards Brownsville, right? And so each of those populations is going to have a distinct collection of not traits so much, but collection of alleles, variations of genes, depending on where those different groups of people originally came from and where their ancestors came from, and so on and so forth. Right. And so really, when we're talking about genetics, we can talk about all of those things, right? And really, what's the, here we go. Really, it boils down to one, hereditary material, right? So this is stuff which is passed down from generation to generation, right? And what does that heritable material do? What does it mean? Right. So really that's kind of the real like core of genetics, right? That hereditary material being DNA, largely, not entirely, but almost entirely DNA, and genes and regulatory structures and mutation rates, things like that. And the traits themselves, right? And so when we talk about trait, and this is a good word to learn, what we mean is the thing that is controlled by that gene or genes. Right, so hair color is a trait, for example. The phenotype is what version of that trait you have. So I have brown hair, so my phenotype is brown for the hair color trait. And most of you will probably have around brown or dark hair, right? Because it's a fairly dominant trait. Also, depending on where you come from and where your ancestors come from. You go to Sweden, and you'll see most people have blonde hair, right? Maybe you've got some redheads. There's one town in Russia, what's it called? I think it's Ossetia, right? 90% redhead. Crazy, right? That is really in large part where redheads come from, if we're talking about evolutionary genetics. I don't know why, but well, I do know there are certain mutations, but why it started there, I have no idea. Right. So, and also how those traits are inherited. Right. What is the genetics of those traits? Right. If we look at one particular trait that you may have or I may have, why is that inherited in a particular way? Right. So really, that's kind of what genetics is all about. And then, and this is also obviously a field of study that has been going on for millennia, right? So in terms of its being called genetics, this is something which is, kind of 150, 170 years old, give or take. Can't remember exactly when Mendel did his P stuff, but I think it's in the mid 1800s, 1860, something like that. But if you think about wheat, actually, let's talk about maize, given our geographical position. Maize is it's a really important food stock. You wouldn't have tortillas or arepas or anything like that without maize, right? So it's a really important food base for Central America. It arose from, and you'll actually see these. I see them every so often when I ride into work. They actually arose from a plant called teosinte, right, which is a kind of like a wild plant you see growing at the sides of the roads around here, certainly more as you go further south, right? But essentially thousands and thousands of years of selection, genetic accidents, more selection has led us to what we now call maize, right? And basically from being, people farming would go, well, this particular plant that's producing nice, big kernels. I'm going to keep some seeds from that. I'll grow that next year, right? And then if you do that over long enough period, and we're really talking about thousands of years here, then you end up with something that you see now, right? And that's pretty cool. So really, genetics in terms of animal and plant breeding has been going on for a very, very long time. Now it's actually called genetics, and now we know a lot more about what drives those particular traits, and so we can be a lot more precise about what we're doing. But really, we're not doing anything too far removed from what those Mesoamerican farmers were doing hundreds and thousands of years ago, which is pretty cool. It's really neat to see it. It's like this kind of big bushy green plant. What's up? Sorry, I don't know your actual name. Is it Zuleika?
Yes.
Oh, cool. Hi, Zuleika. What's up?
Hi. I had a question on where it says the variation within and across taxa. So I looked up what taxa meant, and it said a group of one or more population, so basically a unit. But I don't really understand what you're exactly saying, like variation.
Oh, okay. So essentially, each individual has a collection of.
Within, like,
Sorry, my internet's a bit patchy. Both my kids are hitting it up right now for school. Um, so essentially, each individual has a different collection of alleles, right? We all have the same genes, one hopes, right? We're largely genetically identical, apart from small changes kind of scattered throughout our genome, some of those being in genes, right? And so, as an individual, I'm a little bit different from you, right? Not a lot, just a very little bit, which is why I look a bit different from you. Um, but when you expand that out to a population, right? Now you have uh, basically a collection of alleles. So if you look at one gene in particular, let's say, hair color, right? That's pigment, melanin production, right? Most likely, if you look at the population in San Antonio, you'll have maybe 3 or 4 different versions of that pigment gene, right? Most people have dark hair in San Antonio, by and large, if you look at it that way, right? Now, if you go to coastal California, right, and look at a population there, you'll probably see 1 or 2 of the same versions, but you may see other versions as well, which are not present in San Antonio based on where people are coming from and where their ancestors came from. If you go further east and say look at coastal Mississippi, right, you'll again see maybe 1 or 2 of the same forms of that gene, but you'll see a different, and also the proportion at which those versions exist will change.
okay.
So essentially what we're talking about there is that each group of individuals in a population has its own collection of alleles, and those alleles, that variation in that genetic material, is going to be kind of unique to that population. And then you go look at somewhere else, and it'll be a little bit different, right?
Okay.
And so really, it's trying to get across the point that we're all largely the same, but we're not the same, right? And so how do we differ, between each other? And how do we differ between populations, and how do we differ between species? What are the difference between us and chimpanzees, for example, right? And then you can kind of scale that up as far as you want.
Okay.
So really, when we talk about genetics, a lot of times we're talking about scales, right? Are we talking about, one gene? Are we talking about many genes? Are we talking about one individual? Are we talking about one population, one family, maybe, right? Or we're talking about one species, right? We can kind of, there are a lot of different scales from, small to very, very big, essentially.
Okay. And
So
I
that's
also,
what it means.
sorry to interrupt,
No
but
problem at all. Interrupt away.
okay. I also
Um,
had a question on the mode. Like, what did you mean exactly about?
so really that's what we're going to be getting to in terms of Mendelian inheritance, right? So how do you inherit your traits? Do you inherit them in um, Mendelian fashion? Do you inherit those traits in a non-Mendelian way, which could mean a lot of different things? Um, for example, mitochondrial genes, which control several aspects of metabolism, you get those only from your mother, right? So you can trace your genetic lineage via a matrilineal path through your mitochondrial DNA, which is very much non-Mendelian, right? Because the father doesn't contribute at all, not even one little bit, to your
Mhm.
mitochondrial DNA, right? It's all your mothers and your mother's mothers and your mother's mothers and your mother's mothers, all the way back to, you know, Eve, if you want to look at it that way. But this is really, this PowerPoint is really just to kind of give you the kind of overview. Everything that we're going to be talking about here, we'll be going into in more detail later on in the course.
Okay.
Cool. Good questions, though. Okay, so if we want to talk about this in terms of kind of detail, right? Oh, just got to show you dog in a usual spot on the couch, aren't you? This is really about how do we, well, we can't just say we study genetics, right? I mean, I guess you can. I mean, I'm a geneticist and have been for many, many years. But you need to narrow that down a little bit. So really, I'm a molecular geneticist, you know, but I also use what I call kind of classical transmission genetics to do some of my work as well. I don't tend to, well, actually I did earlier on in my career, looked at evolutionary genetics, right? So really there's lots of different ways in which you can ask the same kind of question. And to be honest, these are really just kind of like lines in the sand, right? Really they are somewhat artificial distinctions. They just make it a little bit easier to talk about particular aspects of genetics. Just the same as you know, biologists will divide up biology into, you know, different fields, botany, developmental biology, ecology, you know, things like that. Just makes it a little bit easier to get a grasp on it rather than just calling it all biology. Right, so really transmission genetics is what we call Mendelian genetics. Well, I mean, it's kind of a little bit of a hamstring, to be honest. Mendel, because everything to do with transmission genetics is now called Mendelian genetics, which is a bit annoying because it's not really truly Mendelian in many ways. But transmission genetics is really about you and another individual reproduce. What's the likelihood of your offspring having a particular trait, a particular phenotype for a particular trait? Whereas molecular genetics really concerns itself much more with you know, how does this gene work, right? How is it controlled? What is its makeup and its structure, right? And between molecular genetics and evolutionary genetics, well, I don't know. Evolutionary genetics can kind of span population genetics, I guess. You could have a bit of an argument with a population geneticist if you wish about that. But really, what we're talking about with evolutionary genetics are populations and time, right? Which is not really something that's considered in transmission and molecular genetics, right? We really, with evolutionary genetics, we're now not just looking at individuals, right? Which is what we care about with transmission genetics, but we're looking at entire populations, right? And most importantly for evolution, how those change over time, right? Because time is really the, you know, the big thing in evolution genetics. Okay, this is a little bit of kind of history. It's actually quite shocking, really, when you think about it, how recently we've started learning this stuff, right? So although this may seem like a long time ago for some of you, it was only in 2000, was it 2001? I can't remember, when the human genome was published, right? Just one of them, right? From, you know, not one individual, I don't think, but just one version of that. And obviously there are many, many versions. And so that's only 20 years ago, which in science is really not a very long time. The other day I watched with my wife a film called Radioactive about Mariam Pierre Curie. That was 150 years ago, give or take. Actually, not quite that long ago. No, 120 years ago, right? Even that is not a super long time ago, right? So really genetics is a pretty young field. And it, to be honest, it really truly kicked off properly with discovery of DNA as the hereditary material, right? And that was in the mid 50s, right? So that was about 70 years ago, give or take. And really, it was probably took off in the mid to late 70s when PCR was invented, the ability to actually amplify selectively genetic material, right? So really, all of the discoveries and benefits of genetics in terms of the food you eat and the drugs you take, and even screening of children, embryos before birth for various congenital diseases and stuff. All of that really came about from in the last 40 years, right? Which is, within my lifetime, essentially. So it's a very young science, not the youngest of sciences, but very young. And so this is actually kind of cool because it gets to a lot of stuff that we'll be learning about later, right? Particularly when we talk about transcription and translation, or the central dogma or gene expression, whatever you want to call it. So it may seem a little crazy, but until the 50s, there was a huge debate about what the hereditary material was, right? Because clearly we knew that traits were inherited, right? That had been laid out by Mendel. It had been rediscovered. Well, that was in the 1850s. Been rediscovered in the early 1900s. But scientists didn't know what that took. It's a little bit shocking to think of that because, it's dogma now, right? DNA is the hereditary material by and large, unless you're a virus, in which case it might be RNA, right? But until the 50s, that really wasn't established. In fact, just as Watson and Crick were, and Rosalind Franklin, and what's that other guy called? There's another one, Rosalind Franklin's supervisor. I can't remember anyway. Maurice something, I think. Just as they were working on their model for the structure of DNA, another group of scientists thought that it was a triple helix, right? Which is kind of crazy. It'd be a little bit like Fifth Element, if anybody's seen that. Super, super cool film. And really, it was the pictures that Rosalind Franklin took, and then essentially the pictures that Watson and Crick kind of stole, which established DNA as the hereditary material and as a double helix, right? And this is really cool because DNA is very simple, right? By that time, we already knew that it consisted of only 4 bases, right? The adenine, cytosine, thymine, and guanine. And so the argument against DNA was that it was too simple. How can you encode all of these many thousands of different proteins, right? The humans is about 20,000 coding genes, give or take, right? So about 20,000 different proteins are encoded by our genome. And so how do you encode all of that complexity with 4 pieces of information, right? And we'll get to how that happens, and you probably know in one way or another how it does already, but we'll get to that later on. And it's really neat, right? That's really the amino acid code, right? The whole magic of translation, essentially, right? And so, oh, there you go, Maurice Wilkins. I knew it was Morris something or other. Actually, it's worth reading up on the story behind this. It's a little bit of a stain on science in many ways, but kind of emblematic of the way world was then, and until fairly recently, to be honest. But essentially, Watson and Crick were, at least one of them is American, were working on this problem, right? And the way in which they studied this problem or scientists at the time, is they would generate crystals of DNA, right? So essentially repeating ordered structures. And then through that crystal, you shine X-rays. Shine, I don't know, you get the idea, right? Electromagnetic radiation. And then those X-rays are diffracted, right? Same way as if you shone light through like a film or grating or something like that, right? Or even just through water, right? The light gets bent. Because the gaps between the different bits of DNA are a lot smaller and kind of on the same range as the wavelength of light, you need smaller waves, right? Or waves with closer peaks, and that's where X-rays come in. And so you shoot X-rays through that crystal, and it will diffract in a particular pattern. I've never understood how they can stare at these patterns and go, "Oh, that's that." You know, it's an absolute mystery to me, but they can. And so the patterns that they got told them that the structure they were looking at had a helical structure, which is what they're pointing at in this picture, and it was a double helix, right? So DNA consisted of two strands, right? Which is kind of what they'd been thinking from some really cool inheritance experiments that have been done prior to this. And so essentially they came across this discovery by collaborating with Maurice Wilkins, who was, where did he work? A large research lab just outside London. Can't remember what it's called right now. And the story goes that Rosalind Franklin was the person who actually took those pictures, right? So she was the one that did the work. But she was at a conference, I think, when Watson and Crick visited Wilkins. And essentially, this is maybe apocryphal, I'm not very sure, saw those pictures on his desk and went, "Oh, wow, that's super cool. Let's check this out." And so essentially, those three got all the credit for that discovery. Whereas the person who actually did the work, whether she would have come to the same conclusions or not is not known, because obviously history went one particular way. It was actually Rosalind Franklin. And just like Marie Curie in the original Nobel Prize for the discovery of, was it radium and polonium, I think? She was not recognized for that discovery. Also, unfortunately, she died from, I think, cancer before that Nobel Prize was awarded. And so that was kind of like a cop-out for the Nobel Prize committee, because they was like, "Oh, well, she's dead. Can't recognize her." But thankfully now it's been, like, the story is much more widely known that it was really these four people that made this discovery, not just Watson and Crick, even though these are the ones that kind of get all the credit for it. Oh, I'll tell you a funny story, a little bit of a tangent. Which one was it? I think it was Watson. I don't know which of these, but basically is turned into a very unpleasant racist bigot who believes that all differences are encoded by genetics, and basically black people are stupid because of genetics, which is, patently not true. But a friend of mine who I knew back in Pennsylvania, she had two cats. And they were these huge, fluffy, I don't really like them very much, but these huge, fluffy cats. One of which was kind of beige color. The other one was black. And so she called them Watson and Crick, because she is a geneticist too. And she called the black one Watson, which always makes me laugh whenever I think about it. Anyway, he's a thoroughly unpleasant person by all accounts, and he's got some very unpleasant beliefs. It kind of seems that people who get Nobel Prizes go one of two ways. One, they think it makes them demigods and they can say whatever the hell they want and they'll be right. And the other way is that they just stay who they are. Unfortunately, Watson went the demigod approach. So again, this is just stuff which you should know already, really, right? It's just good for recap. This it's right-handed, which is kind of neat. It spirals in one particular direction, not the other way. But the bit that is really, really important is this, right? We'll come back to that a lot over the next few weeks. And this is really important because it has lots of consequences in terms of gene expression, in terms of gene regulation, DNA replication, all kinds of different things. And so hopefully by the end of this course, you'll be like, yeah, anti-parallel, that's the one. And you'll be really sure about why that's important. And it's also really interesting because DNA has direction too, right? And that's something that can't be overemphasized. DNA has direction, pardon me. So mRNA has direction. And because mRNA has direction, proteins also have direction, right? So they are not, you can look at polysaccharides, right? Starch, glycogen, what's the other one? Cellulose, right? They do not have a direction, right? One end of the chain, glycogen perhaps not because it's branched, but for starch or cellulose, one end of the chain looks exactly the same as the other end, right? You can't really distinguish between them. But DNA, mRNA, and proteins are different from one end to the other, right? They have a beginning and an end. And that's really, really important. What else have we got? All right, so once you know that DNA is the hereditary material, and you know that it consists of these 4 bases, and really from that point onwards, you know how that information is encoded, right? So we knew what proteins were made up of before we knew that DNA was the hereditary material. But it was only after we discovered that DNA was the hereditary material that we're able to figure out exactly how the information in DNA, right? Because DNA is purely a repository of information. It does nothing else, right? How that information can be turned into something functional, right? Because that's really what we care about. That's what really makes you different from me and from each other. That's what makes, your kind of skin cells different from your tongue cells. Yeah, probably shouldn't have done that. Never mind. And why your liver does certain particular things and does them very, very well. But your muscles are obviously very different from that, right? It's all down to what information is being used in a particular place at a particular time, right? You wouldn't want to do, a marathon just based on the strength of your liver, right? But if you're into Russian drinking games, your liver would be very, very important. Don't get into Russian drinking games. Bad idea. Lots of vodka. Also, one of the things that makes you and I different is that over the course of my genetic history, right, and we're not talking about my parents and my grandparents, we're talking about thousands of generations back, right? So in my case, I'm British, obviously. That's Scotland through my maternal grandfather, mostly. So I'm like a quarter Scottish, give or take. Aye, aye, Macroombe. And about a quarter German. So my paternal grandmother's side, they're from Black Forest in Germany. A lot of you can probably trace your ancestral history kind of all kinds of different places, right? And though essentially what we're talking about is what mutations occurred in those individuals, in the individuals prior to those and prior to those and prior to those, stretching back to, the first Homo erectus maybe, or maybe Homo sapiens that emerged out of Africa, right? And so we are the sum essentially of all of the mutations that occurred in our ancestral lineage, right? And that's one of the reasons why I'm different from you, because my ancestral lineage is different from yours, right? And so for many of you, that ancestral lineage will go stretch back to Central America, right? Some of you, that ancestral lineage will stretch back to Europe, others, you know, who knows where, right? And because those lineages are different, because the mutations that arose, some of them may be restricted to that lineage or to that particular population. That's why we're different. That's why I go out in the sunshine, boom, instantly turn pink, right? I get burnt in like 10 minutes here in San Antonio if I go outside without sunscreen on, because I'm a pasty white Brit. A lot of you go out into the sunshine and you're like, ah, it's freezing out here. It's like 85 degrees. Ugh. And you put like Ugg boots on and coats and beanies and things, which just I find really funny because I'm sweating my ass off. So essentially, that's one of the reasons why we're different, right? Because those changes in in our DNA, although very small, accumulate over very large amounts of time and result in us being different from each other in small but important ways. And also, once you know how those ways, those differences matter, now you can start using that information yourself, right? So if we know that, you know, a particular enzyme produces, you know, starch, or a particular version of that enzyme produces better starch or more starch or a particular type of starch or whatever, right? And we really, really care about that because that's what we're going to be using to create maize flour and tortillas and whatever. Pardon me. Then we can actually take that information and we can stick it into a particular plant for it to have that particular characteristic, right? So essentially, all of that information, all of that knowledge that we've gained over the last 150 years or so, now we can actually start applying that to create things that we want, right? We're not going to be relying on, you know, years and years of selection. Now, if we want plants to be resistant to insects, right, rather than breeding them to be so, we simply take a gene from a bacterium and we get it to be expressed in that plant, right? So whenever insects feed on that plant, they die, right? And so that's BT toxin. Actually, or if you're a Hollywood starlet or star and you're getting a little on the wrinkly side, you can actually have someone inject that into your face and then you'll never be able to smile again, right? And so that's actually the flip side, right? So you can have either BT corn, which some of you may have heard of, right, which comes from the bacterium Bacillus thuringiensis, produces a toxin, creates crystals which disrupt the structure of insects somehow. I'm not totally sure how. Also is commonly known as Botox, right? So that same toxin, if you inject it into people's faces, causes their I think it's like their muscles to like spasm or something. I don't know exactly how it works. And that pulls all of those wrinkles straight. So you look all young and beautiful again. That's a theory anyway. Also means you can tell when people have it because they can't smile very much. And so if we want to take that upper scale, right, we'll take that upper notch, now we can talk about genomics, right? So one of the big things over the last, well, really since the publication of the human genome, one of the really big things, big fields to come out of that is the field of genomics, right? And so before then, really the focus had been on individual genes, right? So, you know, this BT toxin gene, for example, or a starch producing gene, or insulin producing gene or whatever, right? Thinking very much at the small scale. But now that human genome sequence was published, and really that was, it's called a reference sequence, right? So there's an assumption that is made that whatever individual they took that from, that becomes the reference. It doesn't mean that that individual is perfect, right? Because that individual or individuals that they use will have a collection of changes or alleles, which is distinct to that individual or collection of individuals, right? But then every new sequence will be compared to that, right? So essentially what we're talking about with genomics is really the study of as much of an organism's genome as possible, right? So my genome is largely the same as yours, but my genome will have a particular collection of changes which are different to yours, right? And that's why ibuprofen doesn't work for me. I can neck down half a bottle and it will barely make a difference. Right? Whereas other people have to be very, very careful taking ibuprofen because they can overdose on it. It can actually cause liver damage really easily, right? So they can only take, say, one tablet, every 6 or 12 hours. Whereas I could take 20 or 30 and it wouldn't make any difference whatsoever, right? So one of the issues about, and this is where genomics really comes in, one of the issues at looking at a single gene is you may find the one which is responsible for that, right? Maybe, if you're lucky. It's actually very hard to do because most traits are caused by more than one gene. But even if you do, how is that gene controlled, right? What turns it on? What turns it off? What proteins does that protein produced by that gene interact with? What pathway is it in, right? So now when you start asking those questions, the scale starts getting bigger, right? And the other way of thinking about it is we are the sum of our genetic information, right? So for whatever trait you look at, really, that could be caused by many, many different things in terms of genetic information. And so by looking at it at the genomic level, you capture all of that. If you look at a single or even 2 or 3 genes, you're only going to capture part of that information, right? And so that's really where genomics comes in. And that's why it's of such huge interest in the pharmaceutical industry, right? And really, I mean, obviously lots of other people are interested in this too. Evolutionary geneticists are, go gaga over genomics because it actually allows them to look at evolution at a whole genome level, right? Which gives them a lot more insight into what's happening. But in terms of pharmacogenomics, not always, but if you've got really good health insurance, at least, and you diagnose with cancer, one of the first things that they're going to want to do, if you're able to pay for it, is sequence your genome. Because within your genome are going to be a lot of pointers and clues as to how well you're going to respond to different kinds of cancer therapy, right? And so they actually now know that some people are resistant to different kinds of chemotherapy. Other people will be more readily develop resistance to chemotherapy. That's actually one of the main causes of relapse in cancer treatment, right? So you treat with a particular chemotherapeutic, and then after a while, the cancer starts growing back again, right? And so, like, the key now is, okay, we now know everybody is actually different in terms of how they respond to medicines. Some respond very well, some respond too well, right? You can actually overdose at normal doses. Some people don't respond at all. Or they may respond, but after a while, they're more likely to develop resistance to that particular treatment, right? And so by looking at the whole genome of that patient, doctors hope that they can catch a lot more of those problems before they happen, right? So rather than trying you on one particular chemotherapeutic and going, oh shit, that doesn't work, and then they try you on a different one, now the idea is that they'll actually go, okay, this patient is not going to respond to this one, or this one. They'll respond to this one, but we have to be careful with dosage because they might be toxic and so on. So they can build up this picture of what's the best way to treat this patient before ever prescribing a medicine, right? Which is, compared to how it was done before, is like the difference between modern day and the Stone Ages, right? In terms of treatment, right? So essentially, this has just been a kind of a bit of a rambling kind of wander through genetics, but this is really to put all of this into context, right? So we'll be studying a bunch of this stuff. We're not going to be going too deep into genomics because it's a large and very confusing field to kind of tackle. We won't be touching evolutionary genetics at all, unfortunately, but that's because you'll be studying it in evolution, most likely with Dr. Border, maybe next semester or the one after that. But all of the rest of it, we'll be going into it in quite considerable detail, right? So try and keep some of these things in mind as we get really into the weeds, so that you can kind of keep that context and not be overwhelmed by the information that I am throwing at you. And that's pretty much it. So as I think I said last time, there's not a huge amount of stuff in here which you'll need to worry about specifically for revising for exams, but it's a good idea to get the general concepts right, and the language and some of the phrases, phenotypes, traits, genome, things like that. What do those things actually mean? Because we'll be using them a bunch later on. And then on, hang on, let's show you very quickly before you go off. And then on Wednesday, we'll be starting on this PowerPoint, right, which is really some of the kind of background general stuff on the kind of genetics we'll be studying, or some of it at least, right? So have a look at that before Wednesday if you can. And it's, again, still a recap type stuff, but we'll be getting more into the details. And that's pretty much it from me. So have a lovely rest of the day. If you have any questions about anything, let me know. I'll be sending out the lab pairings after this. And so you'll be able to get ready for Wednesday and Thursday if you're in my lab sections. Cool.
Thank you. Have a great day.
You too.
Bye.